Well, it's really a great pleasure to be here with this broad audience on the webinar. And I just want to kind of in the spirit of Bethany's announcement about the uncertainty on the federal land, I just want to say what a pleasure and privilege it is to be part of the NE CASC network. And similarly, you're going to get to hear from three of my colleagues we've all been working together on brook trout and Adirondack lakes, and with support from NE CASC and gaining many benefits from being part of that network that go far beyond the work that you're going to hear about today. So without further ado, Isabella, if you can go to the next slide, that'd be great. So you're going to hear today three vignettes from members of our team. So we work together on a set of about 20 Adirondack lakes and do a lot of muddy boots field ecology in those lakes, where we're looking at the temperature, the oxygen, monitoring the fish populations, and trying to understand how these lakes and their fisheries are changing through time. The four of us that you're going to see, whose faces and voices you'll hear today, are just kind of the tip of the iceberg from this team. So we have a small field station where we have lots and lots of graduate students and undergrads and postdocs that come to work on this set of issues. We also want to acknowledge not only support from the NE CASC, but also our longstanding partnership with New York DEC, in their fisheries office, support from the Cornell Atkinson Center for Sustainability, and to recognize that most of the lakes that we are going to tell you stories about today are ones that lie on private lands. And so we gratefully acknowledge the access to those waters, as well as financial support from those private partners. So now I'm going to tell you a little bit about lakes in the Adirondacks. And so you can see in the backdrop of this picture, and Isabella, if you can give me the next one. So this is the kind of setting where our stories take place. So here we're seeing a lake, this fairly thin soils, a lot of conifers in the landscape up in the Adirondacks, but it's within the Adirondack Park, which is the largest state park in the country. It's about 17% of the state of New York's surface area, and it contains more than 11,000 ponds and lakes. So it's really an amazing landscape for studying lakes. Most of those lakes have water that's rather tea-stained, and that's what you can kind of see in the background of these last two images, was that the water doesn't look nice and clear. You're not seeing the bottom or the biota very clearly, because there's this sort of haze of tea staining. So you're going to hear a lot more about that tea staining from Steve in the first vignette. But it's a hallmark of almost all of these 11,000 lakes and ponds are quite tea stained. They're also fairly low productivity. So these, by virtue of being at a fairly high latitude and then also at a high elevation by Eastern standards, these lakes tend to be fairly cold. So they're temperate lakes. They go from having ice cover in the winter to being quite warm at the surface during the summertime. And those seasonal shifts create big differences in the temperatures and the oxygen that the fish deal with. So next slide, please. So the fish that we want to tell you about are brook trout. Brook trout are the most sought after fish in this region, really throughout the Northeast. Their name, brook trout, would suggest that you find them mostly in flowing waters. Our stories are going to be about brook trout populations that are in fact resident in lakes and ponds. So that's a fairly unusual thing within the range of brook trout. Typically, they are found primarily in flowing waters in rivers and streams. But in upstate New York and the Adirondacks in particular, in parts of southern Canada and in the state of Maine, there are many, many lakes and ponds that contain thriving brook trout populations. And these are a special attraction for anglers. So for over a century and a half, one of the major draws for tourism to the Adirondacks, and it is a tourist economy in the region, one of the major draws has been fishing in lakes for these spectacular fish. So you can see with the white fin edges and beautiful colors on the side, these are not only spectacular fish, but they're also very, very sensitive fish. And so each of the three vignettes that you're going to hear about today are sort of exploring different facets of that sensitivity and the environmental change that these fish are subject to when living in lakes. Next slide. So the first vignette, Steve Jane's going to present, addressing the rate at which environmental conditions are changing and the challenges that poses for cold water fish like brook trout. So these fish require warm, cold water and well oxygenated water. And so Steve's going to tell you about how both temperature and oxygen are changing. Then Connor's going to tell you stories of how the fish respond to those environmental changes. So as the waters become more inhospitable, what physiological and behavioral tricks can they play in order to not just persist, but thrive? And finally, Isabella will tell you about using eDNA to assess loss of brook trout populations from Adirondack lakes. So that's it from me, and I will pass the baton to Steve.
All right, thanks, Pete. So as Pete mentioned, we're working in the Adirondack Park here, and we're really focused in on 16 lakes. Next slide, please. And these are located around the old forage area of the Adirondacks. Next slide. And so as Pete mentioned, brook trout are cold water fish. They also require well oxygenated water. And as surface temperatures are warming, we are seeing declines in deep water oxygen. So this was demonstrated in this paper from 2021 on the left in temperate lakes. And then on the right here, a more recent paper from 2024 showed this using a database of over 8,000 northern lakes. Next slide, please. And on top of this, Adirondack lakes are undergoing a process called browning, which is increased loading of this dissolved organic matter from the surrounding landscape. And this dissolved organic matter adds this tea stain color, as Pete mentioned. Next, please. When we think about what that means for a lake, if we have a clear lake, such as on the left here, light and solar radiation is able to penetrate deep into the water column. And so this allows lots of opportunity for photosynthesis, which generates oxygen, but it also tends to heat those deeper waters. When a lake browns, as you see on the right, that light and solar radiation, it penetrates only to the near surface depths. And so that tends to increase heating right at the surface, but it also decreases heating at depth. And so these brown lakes tend to have larger volumes of this cool water, which could be beneficial for a cold water species like brook trout. But it also shades photosynthesis, which tends to drive down oxygen, and it fuels microbial respiration, which will tend to do the same. Next slide, please. And so this raises this question, will browning ultimately mitigate or exacerbate the effects of warming for cold water species like brook trout? Next slide, please. And so in 2021, we outfitted these 16 lakes with sensor chains of high frequency temperature and dissolved oxygen loggers. So these take readings at like 10-minute intervals for the season. Next slide, please. And that gives us this really high resolution understanding of oxythermal dynamics in these systems. And so if we combine the temperature and the oxygen data, we're able to come up with these habitat suitability heat maps that you see. And when you look at these, the blue generally represents locations in the water column where temperatures are cool enough for brook trout and oxygen concentrations are high enough. The yellow represents locations where temperatures are warm enough that they're going to introduce a physiological heat stress response. It's not necessarily going to kill the fish, but it does cause stress. And then the orange represents temperatures are either too warm or dissolved oxygen is too low. And when we do this, we find that lakes typically fall into one of four categories. So on the far left, we have lakes where at that most restricted point of the season, there's always some of that cold, well-oxygenated water, but it's a relatively small proportion of the water column. These we call constrained. In the center, we have lakes where at the most restricted point, those warm surface waters intrude into those low oxygen conditions. These we call squeezed. And then all the way over on the right, we have lakes where there's very little of this low oxygen water, but there's long periods of time where the entire water column is thermally stressful. And these we call overheated. Next slide, please. And there's one other type of lake. These are large lakes that are generally over 30 meters deep. These lakes always maintain large amounts of this cool, well-oxygenated water. And so therefore they appear to be buffered from both the effects of warming and browning. And so these lakes we refer to as buffered. Next slide, please. And so we wanted to understand the relationship between that dissolved organic matter and these oxythermal conditions. And so in these images here, the x-axis is dissolved organic carbon, or DOC. This is how we measure the amount of this dissolved organic matter in the water column. In the left panel, on the y-axis, we're looking at the maximum depth in the water column where temperatures are greater than or equal to 20 degrees C. In other words, how deep into the water column are those warm, stressful temperatures penetrating? And what we see is if we have a very clear lake, so all the way over to the left, those penetrate quite deep into the water column, so over 6 meters. And we also see that when we brown things a little bit from those really clear depths, that warm temperature boundary shifts toward the surface quite a bit. If we start in a browner lake and brown things a little bit more, that shift is less substantial. So it's a negative exponential relationship. On the right, this is similar, but only now the y-axis is the minimum depth where dissolved oxygen is less than or equal to 5 milligrams per liter. So in other words, how close to the surface is that low oxygen water getting? And again, if it's very clear lake, so over towards the left, it's quite deep, so approaching 10 meters. But unlike temperature, the relationship is the same across DOC. In other words, that shift towards the surface is pretty consistent. It's a linear relationship. Next slide, please. So what that means is if we have a very clear lake and we brown things a little bit, that warm water is going to shift toward the surface a lot relative to how much that low oxygen water shifts toward the surface. So in that case, there is probably going to be actually a little gain of that cool, well-oxygenated water. If we have a browner lake and we brown things a little bit more, the shift of that warm water towards the surface is going to be small relative to the shift of that low oxygen water towards the surface. And then we actually lose that cool, well-oxygenated habitat. Next slide, please. So another way to think of this, if we have DOC on the x-axis here and depth on the y, this brownish line represents that low oxygen boundary, and this green line, that high temperature boundary. If we're starting very clear all the way to the left, we brown things a little bit, we're expanding that really nice habitat. But beyond moderate DOC concentrations, further browning is going to contract it. And if we go out far enough, it actually squeezes it out entirely. And so to illustrate this, this is data from one of our highly monitored lakes. The x-axis here is date through the season. We're looking on the y at depth in the water column. And what we see is we have this huge expanse of low oxygen water at the bottom, and it's intersecting this warm surface water for approximately 2 months. And in fact, this lake does not really hold brook trout very well at all. And so again, next, if we have a little, if we're in a very clear lake, we brown things a little bit, that might be beneficial, but beyond moderate concentrations, it's actually detrimental. So next slide, please. So on the left, this histogram is taken from a data set of over 1,400 Adirondack lakes. These are maximum lake depths. The dashed vertical red line is that 30-meter depth where we consider lakes to be buffered. And we can see that the vast majority of these lakes are shallower than that buffered category. Another way to look at that, in the middle, these bar graphs here, the one on the left is from that same database. We've taken our fitted relationships and applied them to that database to break the lakes up into our categories. The dark green is that buffered category. Next. And we can see that it's a very small number of lakes. It's less than 1% of Adirondack lakes. Now, if we think about those really clear constrained lakes where a little bit of browning might be beneficial, again, applying those relationships, next, please. In the 1980s, that was over 23% of Adirondack lakes. It's now less than 5%, indicating that for most Adirondack lakes, further browning is actually going to decrease that suitable habitat. Next slide, please. And so in summary, browning expands that cool water in a negative exponential fashion. Next. But it expands that oxygen depleted water in a linear fashion. Next. And the net result is that further browning will exacerbate warming in most Adirondack lakes. Next. And so that means that these buffered lakes are critical for conservation of cold water species like brook trout. Next slide. And so that's all I have. And so now Connor is going to talk about how the fish are actually responding to this.
Yeah, so Steve gave a great detailing of the physicochemical changes in these lakes and how these environmental stressors are presumably impacting these brook trout. Now I'm going to provide this sort of fishy perspective. So I'm really interested in how sensitive are these Adirondack brook trout to these oxygen and temperature stressors in the wild? Next slide, please. Okay, so just revisiting some current knowledge, we know that in these Adirondack lakes, these brook trout are experiencing warming and deoxygenation, and this is a presumed negative effect on these populations. And we know this in part because of a number of lab and field studies that have shown that brook trout tend to avoid waters greater than 20 degrees Celsius and will avoid waters with low oxygen levels when concentrations dip below about 5 milligrams per liter on average. But this is an average. Next, please. And this can vary by population and by study. So studies have generally found that their temperature tolerance is relatively consistent, with most field observations noting that brook trout will avoid waters between about 20 and 22 degrees Celsius. Their hypoxia tolerance, however, appears to be slightly more variable, generally avoiding or not tolerating oxygen levels between about 4 and 7 milligrams per liter. And this makes sense considering the fact that there hasn't been a lot of research on hypoxia tolerance in brook trout, and a lot of these methods are somewhat dated now. So the first step was to try and determine, what the specific temperature and oxygen limits are for the Adirondack brook trout. And we could then use these to inform specific Adirondack brook trout habitat suitability criteria. Next, please. The next step was to determine how are brook trout movements being limited by these oxythermal stressors in the environment, and whether or not these movements actually correspond to these lab-generated suitability criteria I just sort of described. And together, next one, the goal was really to identify how are these prolonged oxythermal squeezes impacting wild Adirondack brook trout. Next one. Okay, so the first step, as I alluded to, was trying to determine what are the limits of these Adirondack brook trout. And this involved incrementally increasing the water temperatures that the brook trout experienced. Next one, please. And we found that generally, as we warmed the temperatures, we started to observe negative behavioral responses at about 21 degrees Celsius. So, for instance, we started to notice that their feeding motivation declined significantly beyond this temperature. We also subjected the fish to incrementally declining oxygen concentrations. Next one. And we found that generally their aerobic performance was limited when dissolved oxygen dipped below about 3.5 milligrams per liter of oxygen. So now we've got our kind of upper temperature limits and lower oxygen limits for these Adirondack brook trout that we can then apply to our wild free-swimming brook trout. Next one. Okay, so we've got this criteria. We now want to know what the fish are doing in the wild. To do this, we took a number of these really unique devices, these depth and temperature sensor equipped acoustic telemetry tags that would ping out to a hydrophone receiver in the lake every so often, exactly where in the water column these tagged fish were and what temperatures they were experiencing. In these lakes, we also had, like Steve had sort of described there, an array of temperature and dissolved oxygen loggers so we could accurately assess the temperature and oxygen conditions that the brook trout were experiencing. So in the upper left here, we've got the depth profiles of a number of tagged brook trout over a summer season, with each one of these individual gray dots representing an individual depth measurement. The black line represents the general average trend over that summertime period. Next, please. We can then overlay our habitat suitability criteria that we determined from the lab studies to identify how these fish are occupying, or where in the water column are these fish occupying with respect to oxythermal suitability. And we can see here, generally, it appears that these brook trout are hanging out in this suitable blue band of habitat, as we might predict. And we didn't just do this in one lake, we actually did this in 4 lakes. Next slide, thank you. And we can see that the trend is generally the same. So this is really, really interesting. But we wanted to get a little bit more fine scale and understand how their specific movements and their distributions were being impacted by temperature and oxygen over this period. Next one, please. So the first thing that we wanted to figure out how much of the water column they were using on a daily basis and whether this was constrained during periods of oxythermal compression. And what we found was, as you might expect, when there was lots of habitat, they weren't really constrained at all, and there wasn't really much of a relationship between suitable habitat and daily up and down movements until it got squished down to what I'll call maybe an ecologically important zone, where if there was less than about 2 or 2.5 meters of suitable habitat, we really saw a decline in the amount of vertical habitat they were using on a day-to-day basis. So we can see that during these squeezes, it really is limiting their movements and their use of the water column. Next slide, please. We also wanted to see to what end are their upper and lower limits actually being dictated by these temperature and oxygen limitations that we determined earlier on. And what we found was that, shown in the leftmost figure panel here, when this stressful warmed water got deeper, so too did the fish go deeper. This was also the case for dissolved oxygen, shown in the second panel, or the middle panel. When that stressful low oxygen layer got shallower, the fish also moved shallower. So we can see that their upper and lower levels are indeed being influenced by these oxythermal limits. Next slide, please. Another interesting finding of this research was that the brook trout tended to use cooler water where possible. And generally, we could tell this because their internal temperature recorded by these temperature and depth sensing tags was usually about 2 to 3 degrees cooler than the temperatures recorded from the environmental temperature loggers that were just placed in the lake. And this was really extreme in one of the lakes, Pico Lake, where the fish often were 4 to 7 degrees cooler than we would expect based on the environmental loggers that were placed in the lake, indicating that they probably sought out and found a cool water spring to hang out in for most of the summer, which is very useful to a cool water fish in a hot lake. Next slide, please. And even during these really stressful oxythermal squeezes, denoted by these sort of pink boxes here, we can see that they were able to maintain favorable body temperatures. And in fact, it was often that their body temperatures decreased during these periods, indicating that they probably sought out a really nice chunk of habitat in the lake. But interestingly, if you look really closely here, next one, please, you can see that their body temperature increases dramatically as soon as the squeeze starts to let up and that warmed surface layer starts to dissipate. They also are moving much shallower during this period as well. And what's interesting is when we look at lakes that didn't experience this squeeze, we don't actually see any change in body temperature or a change in depth around this same time. And all this together, I'm taking this as evidence of compensatory foraging, where the fish have been squeezed and constrained of effectively foraging during this period until that upper layer can actually be, just barely suitable enough for them to go and forage. And when it opens up, even though it's still likely stressful for them, they need to make use of that period to try and effectively forage, gain some food, and recoup any energy that was lost while they were stuck in this narrow area of suitable habitat for, in excess of a couple of weeks. Next slide, please. So these results together are suggesting to me that reductions in suitable oxythermal habitat are impacting their behavior. It is impacting their distributions in these lakes, and it's likely causing stress, and it appears to be impairing their foraging, potentially increasing mortality risk for these fish. Next one, please. And we can actually see this in our data and infer it, with a degree of accuracy based on the absence of detections. So these tags are routinely pinging out measurements. So a lack of detections means either the fish has died or been eaten or left the system in some way. Or we can see flatline depth measurements, which really indicates that the fish probably experienced an event of mortality. And these are denoted using these small T's and X's here. And we can see that there's an increased abundance of these events during early heat waves and during these oxythermal squeezes, indicating that there's probably something happening during these periods. So together, I feel like the loss of suitable oxythermal habitat in these lakes is a real issue for these brook trout. And as these oxythermal squeezes become longer and more prevalent, it's likely going to result in greater loss of brook trout and brook trout populations in the Adirondacks, and is definitely something that needs to be taken into consideration and and thought about in more detail. And on that happy note, I'll pass it over to Isabella.
Thanks, Connor. Okay, so now that we have heard a little bit about how lakes are browning and warming, and that's impacting both brook trout habitat and behavior in the Adirondacks, I'm really excited to talk to you a little bit about some preliminary work and future plans for how I use environmental DNA or eDNA to characterize the impact of lake browning and warming on aquatic communities. And so to start out, I'm going to zoom all the way into brook trout genetics and DNA. And when I first started at Cornell, I helped one of our amazing postdocs, Nick Locatelli, generate the whole genome sequences of approximately 200 individual brook trout. And these individuals were representative of nearly the entire native range of the fish species, as well as 6 strains that we stock specifically in our program at Cornell. And this was widely the result of collaborations across the northeastern United States and southeastern Canada, who sent us literally hundreds of fin clips and DNA extracts to be able to do this work. So to them, we are very grateful. And when we generated these genomes, we were really interested in looking at the genetic diversity between stocked and wild populations, as well as we were thinking a lot about using eDNA as a tool to distinguish between the 6 strains, hopefully to be able to then assess profiles in stocked lakes. And as we worked specifically with the mitochondrial genomes, because that's what we typically use in eDNA, we saw really high diversity across the genomes, which is really interesting. And unfortunately, that diversity didn't necessarily align with the 6 strains that we were hoping to distinguish between. But we were able to find 2 gene regions that, when amplified together, could be used to discriminate between 3 strains using eDNA. And this is really exciting for us because it opens the door to potentially be able to monitor strain portfolios over time in stocked lakes, which is something we're really interested in. And so to be able to test this, I'm going to zoom out now to tell you a little bit about a full lake manipulation experiment that I ran. And for this experiment, I stocked the 3 distinguishable strains in 3 different cages in Combs Lake. And Combs Lake is a very shallow and lake with pretty high DOC concentrations, meaning throughout the summer it gets very warm and quite hypoxic, and brook trout cannot survive. And so to do this, like I said, stocked the 3 different strains in 3 cages in varying quantities. And I proceeded to develop a series of manipulation configuration changes in the experiment. And that included removing fish entirely from the lake, as well as releasing them into the lake at different periods of time. And I carried out these configuration changes to accomplish 3 main objectives. One was to simply observe the eDNA degradation spatially and temporally in a natural setting. 2 was to test this ability to distinguish between the 3 strains using eDNA alone. And then the last one is something I'm starting to sample for now as the summer months come upon us, and that is to try and detect population extirpation in a lake ecosystem. And I couldn't move forward talking about this without showing you this very impressive photo of fluorescein dye, because in addition to simply collecting eDNA samples, so just collecting a water sample and hoping I had target DNA in there, I also wanted to make sure the water was flowing at a rate and in the correct direction of my sampling plan that matched with my configuration changes. And based on the fluorescein data, I can confirm that it was. And so moving forward now, I'm still processing the samples in the lab, and so I can't share with you real-time data, but something we might expect to see based on the configuration changes that I made can be shown in this little mock-up that I made of what we might find in our data. And so looking at the x-axis, which is showing us simply time, so the samples that I collected over time, and then the y-axis, which is telling us about the number of samples that we would detect these different strains in. And so we might see something like this, where for windfall, because we had double the number of fish in this cage compared to the other 2 strains, we might see more samples detecting that strain's DNA than the other 2, where the other 2 we see less. But I did remove the windfall fish entirely from the lake about halfway through the fall sampling. And so we might see reduced number of samples, a quick reduction in the number of samples we're detecting those species, that strain in. And then moving forward, as I move through the summer and I sample as the lake heats up and becomes hypoxic, we might see something like this if those 3 strains exhibit different resiliency toward changing conditions. And so if we think about this 3rd objective, detecting population extirpation, what we might see is that if we had a resilient strain or 2 in that lake, as the summer months heat up and our lake heats up, we are not going to see a big change in the number of samples we're detecting the strains in. But if we had a more sensitive strain to these changes, what we might see as the lake heats up and those fish either leave the system or perish is a rapid reduction in the number of samples we're detecting that strain in. And that's simply because as they're no longer in the system, they stop shedding their DNA in the water column, and I can no longer collect it in my samples. And so now, to take us one more step back at the system level, I'm excited to tell you about how I plan to use these methods that I'm currently testing to look at these trends in the Adirondack Park. And lucky for me, the Adirondacks is a great place to do this because it has a long history of long-term and/or large-scale studies of lakes. And some examples of this are the Adirondack Effects Assessment Program, which studied 28 lakes over the course of 20 years, and the Adirondack Lake Survey, which took the opposite approach, studying more than 1,400 lakes over the course of 3 years. And one last example of this is a multi-institutional state-funded project that my lab group is currently involved in called the Survey of Climate and Adirondack Lake Ecosystems, which is where we're taking a similar approach to the Adirondack Lake Survey, and we're going to be studying actually a subset of those lakes over the next few years. And for this big project scale, we're going to be collecting data using a variety of methods, one of which I'm really excited about, obviously, is environmental DNA. But the other one that I think is going to be really cool is remote sensing data. And what I'm really excited about for remote sensing data is that you can actually see lake browning and warming from space using satellites. And what's awesome about it is that we can date this information back all the way to 1984 when Landsat 5 was first launched, which conveniently aligns very well with when the Adirondack Lake Survey began. And then it can also extend that information all the way to present day and moving forward as satellites continue to collect this data. So that allows us to not just have this information about DOC concentrations at random time points when someone decided to collect a sample, but it tells us detailed information about the initial state of the lake, final state of the lake as we know it today, as well as a very detailed rate of change over time. So if we pretend like I have pulled this satellite data and I've processed it in a way so we have these brownness values or DOC concentrations, I pulled some data from the AEAP survey that happened from the 1990s to the 2010s to give you an example of what I'm hoping to do with this data. And so you can see here I pulled data from 3 different lakes, all of which have showed a significant increase in DOC over the 20-year study. But you can also notice that all 3 of them have very different rates of change as well as different starting and ending points in terms of their concentrations. And if we use those values, and we think about that in the context of what Steve told us about, that DOC is actually a really good predictor of suitable habitat, we can then use this information to predict the available habitat for brook trout over this period of time. And if we then think about that in the context of what Connor has told us, that there are these thresholds of habitat availability that brook trout need to not just survive, but to thrive, we can look at these lakes and kind of get an idea of where they're doing well and maybe where they were struggling or can't survive at all. So when you think about this in terms of Limekiln Lake here at the bottom, we see that throughout the whole course of the AEAP study over those 20 years, they had quite a bit of suitable habitat. And we can tell this because the green line here at the bottom is our DO threshold, above which we have enough oxygen in the lake. And below this blue line right here is our temperature threshold, below which we have cold enough water for the brook trout to survive. Now, if we think about that for Sagamore Lake right here, we see that while there were periods of time when the DOC concentrations were lower and there was suitable habitat sufficient enough for them to thrive, we also see these peaks here in DOC that align and correlate with these really compressed habitat areas where they would likely be really stressed. Now, if we take that one more step further to Long Lake, which has even higher DOC concentrations, you might notice that it looks a little different. And that's because the area where the water is cold enough for brook trout to survive below this blue line does not overlap at any time point with the area where there's enough oxygen above the green line. So now if we think about this in the context of where the fish are actually living and what communities are present, we can pull data from the ALS survey that happened in the 80s, and then scale, which we're going to start collecting now. And we can look at the change in the community composition compared to the DOC initial values and final values, as well as the rate of change, to understand what is having a bigger impact on these communities. This is something that I'm really excited about and also think is really important because as we move into a warmer world, I think that understanding how culturally and economically important species, such as the brook trout and the Adirondacks, are responding to these different pressures is going to really help us manage and protect systems in a way that maintains spaces where they can continue to live. And with that, I'll pass it on to Pete.
Thanks, Isabella. So that concludes our 3 vignettes. So just to kind of sum up here, what Steve showed you is that we've now conclusively demonstrated that these oxythermal squeezes are eliminating habitat for cold water fish species in many lakes. That's not to say that the fate of all Adirondack brook trout is sealed and that they're all going to come to to absolute extirpation or at least near demise in the next few years. But the future doesn't look good, and many lakes that historically have held brook trout are unlikely to be able to support them in the future. Connor showed you that brook trout are remarkably flexible in their physiology and behavior, and that gives rise to a lot of endurance. And so despite the fact that these lakes are becoming increasingly inhospitable to cold water fish like brook trout, they still have some tricks up their sleeve that may enable them to hang on, even in places that we don't think that they should be able to. And then Isabella told you about novel uses of both eDNA and remote sensing that she's using to explore which lakes are losing their brook trout, how sensitive is eDNA to that, and can we use remote sensing to look at a landscape scale to identify lakes that over the last 40 years have changed rapidly enough that we should really be concerned about their capacity to hold on to the trout. Next slide, please. So just to close things out here, because we want to leave time for a question and answer session, I do just want to mention a big element of our partnership with New York Department of Environmental Conservation is on a project called SCALE. SCALE is a big consortium of research institutions, both universities and also nonprofits throughout New York State that are coming together to survey many lakes of the Adirondack. So we're targeting about 400 lakes to be surveyed in the next couple of years. We've done pilot work in 2023 and 2024 that's setting the stage for the kinds of landscape inferences that Isabella was talking about. So that's including remote sensing, including eDNA, including food web analyses, lots of physics and chemistry. So as a team, researchers of New York State, in partnership with DEC, the state agency, are trying to really figure out and benchmarking against the 1984 to 1987 Adirondack Lake Survey, how fast have lakes changed? Can we predict which ones are most likely to lose their cold water fish, such as brook trout? And conversely, how fast are warm water species invading these lakes? Because we know that this is one of the huge challenges in this landscape is the arrival of smallmouth bass and golden shiners and many other warm water fish that don't belong in Adirondack lakes. So as those lakes get warmer, we expect to see more of those invasions. And so by testing the rate of invasion as well as the rate of loss of cold water species, we'll be able to anticipate which lakes are most at risk and target those for interventions. I want to just close by mentioning one other partnership with New York DEC, which was kind of captured in some of Connor's slides, that we're trying to evaluate which particular genetic strains of brook trout that are already in the hatchery system. So these are the 6 strains that Isabella mentioned that we've done the mitochondrial genome analysis of. Which of those are most resilient to warm temperatures or to low oxygen? And then to analyze whether stocking programs could be tweaked in order to respond adaptively to climate change by, we're not quite building a better brook trout, but we can be smart about which strains we put into which lakes and perhaps using portfolios, multiple strains that together build resilience into our stocked fisheries. So given that that's already a huge focus for maintaining the cultural and recreational and economic value of this emblematic fish species, we think that we can probably do better with our stocking efforts down the road through climate adaptive stocking.